Hello friends, today I came with a new video on the topic DNA, which means deoxyribonucleic acid. We know nucleic acid or nucleotide consists of three things: sugar, sugar, phosphate, and a base. Sugar molecule can be ribose and can be deoxyribose. In RNA, it is ribose. It is a 5-carbon sugar. That is, we will make a 5-carbon sugar, have H on one end and OH on the another. This is first carbon, second carbon, third carbon, fourth carbon, and a fifth carbon. So, in ribose, second position is taken by the OH molecule, but in deoxyribose, it is only H, which means deoxy means removal of oxygen. Next thing is a phosphate, and third one is the base, which consists of two components, that is purine and pyrimidine. Purines contain adenine and guanine. It is denoted as A and guanine is denoted as G. And pyrimidine are cytosine and thiamine. Cytosine is denoted as C, thiamine is denoted as T. But in the RNA, there is a third pyrimidine known as uracil, which is denoted by U. Now, in the DNA structure, which is a double helical structure, in this, sugar and phosphate form the backbone of the DNA molecule. This is the backbone, and adenine or another bases come inside it. Likewise, this C, T, C, T. Adenine will base pair with thiamine, and thiamine will base pair with adenine. Guanine will base pair with cytosine, or cytosine will base pair with guanine. It means adenine will form a base pairing with thiamine on the another strand, and this bond is a double bond made up of hydrogen, and between G and C there is a triple hydrogen bonding. It consists of a diameter 20 angstrom, and one turn consists of only 10 nucleotides, covering a distance of 34 angstrom, having 3.4 angstrom distance between two nucleotides. So, here is DNA. It is formed of two things. One is a histone molecule, which is a protein, and another one is the DNA and histone. It forms the chromosome, which is present inside the nucleus in eukaryotes, but in the prokaryotes it is not present inside the nucleus like in bacteria. It is just spreaded inside the cytoplasm. So this is known as nucleoid. But in the eukaryotes, which consist of plants and animal cell, DNA or a chromosome is present inside the nucleus. Chromosome, which is made up of DNA. It is present inside the nucleus. But in the eukaryotes, DNA is also present in the semi-autonomous organelles like mitochondria, and another one in the plant cell, it is chloroplast also, which consists of the nucleoid, not the nucleus in which chromosome is present. It is present in a thread-like structure in the cytoplasm in the form of the nucleoid. So we were discussing about that histone, DNA is made up of histone, chromosome is made up of histone and the DNA. So in this, histone is H1 molecule, H2A, H2B, H3 and H4. These four form 8 bowl-like structure like this, like H2A, H2A, H2B. It can be present anywhere, H3, H4. But it forms a 8 bowl-like structure, which is covered by the DNA molecule. This is the DNA molecule and H1 histone. It is again formed after some distance. It is coiled. But these two structures, this structure known as the nucleosome, is again formed after some distance, 8 bowl-like structure, which is linked by the linker, DNA. In this, if DNA is coiled to chromosome, it get coiled, nucleosome structures get coiled around themselves like this. These are the nucleosomes. These are coiled and thus form a chromatin, which if it is further coiled like this, it forms a chromosome structure. Which is joined at some point. This is the coiled structure of the DNA, which is formed by these histone proteins. So in some region, it can be loosely coiled, or in some region it can be tightly coiled. Loosely coiled regions are known as the euchromatin, and tightly coiled regions are known as the heterochromatin. For this we can take an example of the thread, like कोई धागा लेते हैं, उसको spread करते हैं, ठीक है. तो ये जो धागे वाली structure है, इसको हम बोल देते हैं chromatin thread. जब इसको किसी चीज के ऊपर रोल करने लग जाते हैं हम, लाइकवाइज इसके ऊपर ही अगर हम रोल करने लग गए उस धागे को, तो इट विल फॉर्म दिस टाइप ऑफ स्ट्रक्चर विच इज नोन एज अ क्रोमैटिन। जब हम ऐसे ही दो क्रोमैटिन को जोड़ देंगे कहीं से तो एक क्रोमोसोम बन जाएगा जो किसी पोजीशन से अटैच होगा, जिसको हम पूरे को क्रोमोसोम बोलेंगे। और इसमें से कुछ ऐसा पार्ट होगा जहाँ पे थ्रेड हमने यहाँ पे राउंड ऑफ करा, कुछ पार्ट में थ्रेड बहुत ज्यादा आ जाएगा, कुछ पार्ट में बहुत लूजली आएगा। तो लूज वाले पार्ट को हम यूक्रोमैटिन बोलेंगे और जो टाइटली पैकिंग होगी उसको हम हेटेरोक्रोमैटिन बोलेंगे। सो थैंक्स। अगर आपको मेरी वीडियो पसंद आई तो प्लीज़ लाइक करें और सब्सक्राइब करें हमारे चैनल को।